I'm Charles Wyckoff, retina specialist, Houston, Texas, and it is an absolute privilege and a rare treat to be here with two good friends and movers and shakers in our space, Carmen Puliafito from California and Cedric Francois, CEO of Apellis Pharmaceuticals. Great to be here with both of you.
Thank you so much, Charlie.
Thank you, Charlie.
So let's jump right in. So here we are, August 10th, 2023. We're on the heels of ASRS. And let's go through. There was a lot to unpack from ASRS all across retina, not just related to Apellis, but we'll focus on Apellis and some of the data around Pegcetacoplan here tonight. And Cedric, let's start with OIS, right? That's how ASRS gets started every year. You were given the OIS Innovator Award, an important recognition in our field for moving the retina field forward. Congratulations. Tell us about that.
Well, it was very humbling to me. I mean, I have to say, I didn't expect it. I felt incredibly grateful. But to be honest, I don't feel that like a very personal award. I mean, there are so many people that were involved in the development of SYFOVRE. So much hard work over 2 decades. It was a wonderful recognition that I feel I share, not just with the people at Apellis, but with, quite frankly, the whole retina community as well.
Very well deserved. You've been pioneering complement science for many years. So let's move right into a discussion about Pegcetacoplan SYFOVRE. Right, there's been obviously a lot of discussion around the events of retinal vasculitis and intraocular inflammation reported in real-world utilization of SYFOVRE. Tell us about that. Tell us where we are.
Yeah. So first of all, when you hear about these cases, of course, we take them very seriously. When they happen, it's terrible for the patients. It's terrible for the physicians that treat these patients. And each of these cases, we take particular care of trying to understand them and looking deep into them. In the real world, since the launch in March, we have distributed approximately 68,000 vials into the real world. And we estimate, and we can estimate that kind of based on the usage and the inventory management, that about 60,000 of those were actually administered to patients. And we had these very rare events of, retinal vasculitis, sometimes occlusive, sometimes not, associated with intraocular inflammation, where the most important question that we asked ourselves the second that we found out about these was, is this something that is increasing over time, or is this something that is sporadic in nature? And is there a rate that we can understand behind these events? So in that sense, the ASRS, notice that was sent out was very good because it really turned over every stone in the country to try to understand what is going on. So what we had was 7 confirmed cases, and then 1 case that we are still looking into. We had 2 cases in April, 2 in May, and 3 in June. Our task now is to try to understand the nature of these events, why they happened, and all that is, of course, going to be subject to a deep investigation. It is worth noting that these cases happened after a first injection. The reason why I mention that is that, the concept of immunity against the drug, which is, of course, something that everyone is rightfully concerned about, would imply that there is pre-existing immunity at work. And that is something that is, hard to imagine with a drug that no one has ever been exposed to. So, more work to be done, but, that was an important first observation.
Fascinating.
What are the next steps in the investigation?
So the very first thing that we wanted to do, Carmen, was to find out whether manufacturing had somehow changed, if the specs were affected. So we went and did a thorough investigation of the product that we shipped to physicians and compared it to our clinical trials, right? Because in our clinical trials, as you know, we had an enormous body of work. We did 23,000 injections through all of the clinical trial work that we did. And this never happened in our clinical trials. So the very first question was, is the product that we are shipping to physicians today different from the product that we used in the clinical trials? And the answer is no. So now we are going through several other steps. And, we're not going to go in detail through that because, quite frankly, we'd like to communicate when we actually know and have something, right? But there are several leads that we are looking into to try to understand this better. But the common denominator in all of this, in all of these efforts, is again the stark difference between 23,000 injections in a well-controlled environment, and then these north of 60,000 injections in the real world, where all of a sudden we saw this.
Appreciate that clarification. And looking then back to the clinical trial data that you just brought up, right? 23,000 injections. I think that includes patients in the GALE extension study. How are we doing with that data? Remind us how long that extension trial is. Where are we, and what have we seen so far?
Yeah, so GALE is something that I'm personally very proud of. It was a huge investment for us to make, where we decided that the patients that would get to the end of the Derby and Oaks trials would have the opportunity to continue in a 3-year extension trial. And so we have right now patients that have been on 5 full years of dosing already with SYFOVRE. And it allows us, of course, to continue to monitor safety, but also to evaluate the efficacy over time. Because one of the most appealing features of SYFOVRE is that the longer you treat, the more you slow down the lesion growth. That was very evident already in the 2 years of the Derby and Oaks study, and is of course part of our label. So the real question was, in that GALE extension study, we are now with all patients beyond the 30-month time point. What is the efficacy from month 24 to 30? And we had a wonderful presentation at the ASRS by Dr. Steiny that was given around that. But I'll give you one metric that definitely resonates a lot with us. In patients with non-subfoveal lesions, so with extrafoveal lesions, in other words, I don't need to tell you the patients where most vision can probably be preserved, the slowdown for monthly injected patients was 45% compared to the extension of the sham that we had. So those are very large numbers and very important slowdowns of the lesion growth that we believe will greatly benefit patients if they stay on treatment long enough.
Cedric, I think the field is fascinated by this observation of increasing treatment benefit over time. It's fascinating to me personally, having seen various drugs coming, go through FDA approval process, that's in the package insert. This increasing treatment effect is in the SYFOVRE package insert. It's meaningful. I think it's meaningful to the FDA and it's meaningful to the field. What is it telling us about the biology? Do we see this in other neurodegenerative diseases? What does this mean to you?
Yeah, thank you, Charlie. This is by far the most exciting thing that we have observed with SYFOVRE, right? And it is something that is incredibly rare, right? I mean, we're all physicians. When we think about which drugs do we know that start working better the longer you treat, I mean, you have to think really carefully, right? Yeah. But it's of course something that, if you're a patient with geographic atrophy, you're going to be on treatment for many years, is going to be really important in terms of understanding the benefit that you can gain. As it relates to the biology behind it, what I'm going to say now is a hypothesis, but I think one that does make sense. When you have a patient and you treat that patient with SYFOVRE, the cells that sit around the lesion are cells that are visible on your autofluorescence, i.e. they're there still, but they are very ill, right? And many of these cells are probably at a stage where they no longer can recover. Now, when you start treating these patients and the lesions grow over time, 1 year later, the cells that are now at the border have already been exposed to the drug for a year and may therefore be more resilient to cell death. And I think what's important to point out in this context as well is that what we measure with autofluorescence are retinal pigmented epithelial cells, not the photoreceptor cells. And one of the facts that we talked about at the SRS, which I find an absolutely fascinating biological observation, is that the protective effect on the photoreceptors with SYFOVRE looks like it is complete, 100% from the first time of treatment. And you could say, well, how does that match together? Well, what we now know, and we, with the segmentation that we can do with artificial intelligence, we can actually separate the RPE layer from the photoreceptor cell layer. And what we know now is that the photoreceptor cells are the first ones to die, and the RPE follows later. And when I speak to layman people, I always explain it with a sod analogy. I'm like, if you have a lawn of grass, think of it as having a hole in the lawn, and then an area where you only have soil, and then an area where you have soil with grass, right? And when we start treating with SYFOVRE, what we see is that the grass stops dying immediately with the first injection of SYFOVRE that you give. The soil is the one that continues to expand with increasing effects over time. And when the soil reaches the grass, then the grass, the photoreceptors, die in conjunction with the RPE. And then, you see that very clearly. And then they both ultimately slow down to, hopefully very meaningful effects over time.
Fascinating description, and I really have enjoyed reading and learning from Frank Holt, Ursula Schmidt. They both independently done some fascinating work. I think sort of demonstrating what you're describing. Both great manuscripts that they've published that are really teaching us a lot. Thank you for leading that for the field. So, but let me pressure test that. What about visual function? So if we're seeing all this preservation of photoreceptors, we saw the microperimetry data there at the end of the Oaks trial, where there was a signal. I believe that signal was real when you looked in that perilesional zone. What do you think about functional data?
Yeah, so this is also very exciting work, right? And very important work. First off, the reason why we take lesion size or growth reductions as a primary endpoint in geographic atrophy is because visual acuity, as we all know, is not full visual function, but really foveal acuity. Right? And since there is foveal sparing in geographic atrophy, if you wanted to really look at visual acuity in GA, you would have to take patients with GA that has not involved the fovea yet is close enough to the fovea and growing in the direction of the fovea to implicate the fovea. And that, if you had the ability to enroll patients with that particular phenotype, then of course a VA study would make a lot more sense. When you look at overall VA in a GA study, you get a lot of noise, right? You have patients where the fovea is already involved, which was 60% of the patients in our trials. You have patients where the lesion is too far away from the fovea to ever impact it, right? But those VA numbers in the overall picture get included. So what we did in the post-hoc setting is we said, what if we now look at patients that have a lesion that is close enough to the fovea to implicate the fovea? And when you do that, i.e. patients that arguably, at least on visual acuity, have the most to gain, we saw 5.6 letters of improvement in the active versus the sham control arm after 2 years of dosing. So a little bit north of 1 line. The other assessment that we did, and again, this is one, you all know how labor intensive it is for you and for the patients to do microperimetry. So we have this vast trove of data where in the Oaks study, which was 600 patients, right? I mean, we did these microperimetry analyses every 6 months to make sure that we could track what was happening over time. And what we found there was that, again, with microperimetry, you have 68 points. If you look at all the points, you get a lot of noise. I mean, points that are sitting in the scotomatous zone are not going to come back. Points that sit too far away from the border of the lesion are not going to change either. So what we did is take a refined analysis and look at the zone around the lesion and look at the points there. And both for scotomatous points as well as sensitivity, we saw a clear difference between the treated and the sham control. And again, very exciting to me, the same trend towards an increased effect over time, the same way we saw it with the lesion size reduction.
Is there a strategy for using artificial intelligence or other means to document the preservation of retina and photoreceptors or limiting lesion growth?
Absolutely, Carmen. And that is one of the, so we presented data on that at the ASRS. So one of the things we did in Gale, we first had seen this in Derby and Oaks in a post-hoc setting. And then in Gale, we had the opportunity to take the sham control patients. And after 2 years in Derby and Oaks, those sham control patients went on active in Gale. And in a prospective fashion, so, pre-specified and defined, we were now able to look at photoreceptor cell loss in patients with 2 years of history and no treatment. And that's where we saw in the first 6 months of treatment in the GALE extension study that the photoreceptor cells were 100% preserved. And that is going back to what we discussed earlier. After 6 months, the RPE cells catch up with the photoreceptors, and then they start, unfortunately, still dying in conjunction with each other now, right? To ultimately then slow down to these very meaningful numbers when you get to multiple years of dosing.
Fascinating. You know, there's a lot more data to come. You know, these challenges in the real world are important for the field. I'm glad that your team has a strategy to move us forward. You know, I know that many physicians out there are continuing to use SYFOVRE and are excited for the opportunity to treat these patients, and are also looking for more data from you, both from real world data as well as additional clinical trial data. Anything else you'd like to add, Cedric or Dr. Puliafito?
Carmen?
Where do you see the underlying biology of the safety issues going?
You know, I'm hesitant to speculate, Carmen. We, of course, have a lot of hypotheses that we are diving into, and we take this very seriously. And we have, thankfully, an enormous database to work against, right? From these clinical trials. So, you know, like in all these situations, I think either we're going to find something in the very near future that we can hone in on, or, and I hate to say it, but sometimes, you know, these things you can never figure out. So I think the next month to 2 months are going to be very critical, where we can check off kind of the important and most evident boxes. If that doesn't do it, then at that point in time, I think the rate, the rarity of these events, the sporadic nature of these events becomes something important, and we will provide as much information as we can to understand that rate, so that you as physicians can then discuss with your patients whether the risk benefit is something that they are willing to engage in.
The transparency of Apellis and the chief medical officer, Dr. Caroline Baumel, was publicly acknowledged at the ASRS, and we'd like to recognize that as well.
Thank you, Carmen. I have to say, this has been very difficult for Caroline, as you can imagine, right? As our freshly minted chief medical officer, I don't know what we would do without her. I mean, she has been extraordinary. She has the highest ethical standards, understands, of course, these cases very well, and trained with the best, right, Carmen? So that is, we're very fortunate to have her.
Yeah.
Yeah.
Thank you for moving our field forward. We look forward to additional data. Thank you very much for the conversation, Cedric.
Thank you.
Thank you so much, Charlie. Thank you, Carmen.